Chapter 9 Biomolecules. There is a wide diversity in the living organisms in our biosphere. Now, a question that arises in our minds is: are all living organisms made up of the same chemicals, that is, elements and compounds? You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others, and their respective contents per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust as an example of a non-living matter, we obtain a similar list. What are the differences between two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of Earth's crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to element is higher in any living organisms than in the Earth's crust. Table 9.1. 9.1 How to analyze chemical composition. We can continue asking in the same way what type of organic compound are found in a living organisms. How does one go about finding the answer? To get the answer, one has to perform the chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in a trichloroacetic acid Cl3 CCOOH using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through the cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically the acid soluble pool, and the second retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify the particular organic compound. It will suffice to say here that one extracts the compounds, then subject the extract to various separation techniques till one has separated a compound from all the other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to the compound, gives us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from the living tissues can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to done. One weighs a small amount of living tissue, say a leaf or a liver, and this is called wet, and dry it. All the water evaporates, the remaining material gives a dry wet. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that means CO2 and water vapor, and are removed. What is remaining is called ash. This ash contains the inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of the living tissues in the form of hydrogen, water, chlorine, carbon, etc., while analysis for the compounds gives an idea of the kind of organic and inorganic constituents present in the living tissues. From the chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into the acid, nucleotide bases, fatty acids, etc. Amino acids are the organic compounds containing an amino group and an acidic group as a substituents on the same carbon, that is alpha carbon. Hence they are called alpha amino acids. They are substituted methanes. There are four substituent groups occupying the four valency positions. These are hydrogen, carboxyl group, amino group, and a variable group designed as R group. Based on the nature of R group, there are many amino acids. However, those which occur in the proteins are only of the 20 types. The R group is in these proteinaceous amino acids could be hydrogen, means the amino acid is called glycine, the methyl group alanine, hydroxymethyl, serine, etc. 3 of the 20 are shown in the figure 9.1. The chemical and the physical properties of the amino acids are essentially of amino, carboxyl and the R functional groups. Based on the number of amino and the carboxyl groups, there are acidic, example glutamic acid, basic, example lysine, and neutral means valine amino acids. Similarly, there are aromatic amino acids, that is tyrosine, phenylalanine, tryptophan. A particular property of amino acids is ionizable nature of NH2 and COOH groups. Hence, in the solution of a different pH, the structure of amino acid changes. Lipids are generally water soluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be methyl means CH3 or ethyl C2H5 or a higher number of CH2 groups. 1 carbon to 19 carbons. For example, palmitic acid has the 16 carbons including carboxyl carbon. Arachidonic acid has 20 atoms including carboxyl carbon. Fatty acid could be saturated means without double bond or unsaturated with one or more carbon-carbon double bonds. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here are the fatty acids are found in esterified with the glycerol. They can be then monoglycerides, diglycerides and triglycerides. These are also called fats and oils based on a melting point. Oils have lower melting point, example gingerly oil, and hence remain as a oil in a winters. Can you identify a fat from a market? Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. They are found in the cell membrane. Lecithin is a one example. Some tissues, especially the neural tissues, have lipids in the more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are the nitrogenous bases adenine, guanine, cytosine, uracil and thiamine. When found attached to the sugar, they are called the nucleosides. If the phosphate group is also found esterified to the sugar, they are called the nucleotides. Adenosine, guanosine, thymidine, uridine and cytidine are the nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, cytidyllic acid are the nucleotides. Nucleic acid like DNA and RNA consist of a nucleotides only. DNA and RNA function as a genetic material. 9.2 Primary and the secondary metabolites. The most exciting aspect of the chemistry deals with isolating thousands of compounds, small and big, from the living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugars, etc. For reasons that are given in the section 9.10, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of the compounds shown in the figure 9.1. These are primary metabolites. However, when one analyzes plant, fungal and microbial cells, one would see the thousands of compounds other than these primary metabolites. Example, alkaloids, flavonoids, rubber, essential oils, antibiotics, color pigments, scents, gums, spices. These are all called secondary metabolites, table 9.3. While primary metabolites have the identifiable functions and play known roles in the normal physiological processes, we do not at the moment understand the role of functions of all the secondary metabolites in a host organisms. However, many of them are useful to human welfare, example, rubber, drugs, spices, scents and pigments. Some secondary metabolites have the ecological importance. In the latter chapters and years, you will learn more about this. 9.3 Biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have the molecular weights ranging from 18 to around 800 Daltons DA approximately. The acid insoluble fraction has only four types of organic compounds, that is proteins, nucleic acids, polysaccharides and lipids. These classes of compounds with an exception of lipids have the molecular weights in the biomolecules in the range of 10,000 Daltons and above. For this very reason, biomolecules, that is chemical compounds found in a living organisms, are two types. One, those which have the molecular weight less than the 1000 Dalton and are usually referred as to the micromolecules or simply biomolecules, while those which are found in acid insoluble fraction are called the macromolecules or biomacromolecules. The molecules in the insoluble fraction, which the exception of lipids, are polymeric substances. Then why do lipids whose molecular weights do not exceed 800 Dalton comes under acid insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight compounds are the present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane has the another membranes are broken into pieces and the form of vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicle get separated along with the acid insoluble pools and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together they represent the entire chemical composition of a living tissues or organism. In summary, if we represent the chemical composition of a living tissue from the abundance point of view and arrange them class wise, we observe that the water is the most abundant chemical in the living organisms. Table 9.4 shows us that average composition of cells, water 70 to 90% of the total cellular mass, proteins 10 to 15%, carbohydrates 3%, lipids 2%, nucleic acid 5 to 7% and ions 1%. 9.4 Proteins. Proteins are the polypeptides. They are linear chains of the amino acids linked by a peptide bonds as shown in the figure 9.3. Each protein is a polymer of an amino acid. As there are 20 types of amino acids, for example alanine, cytine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is important as the later in your nutrition lessons you will learn that the certain amino acids are essential for our health and they have to supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make while we get the essential amino acids through our diet or food. Proteins carry out too many functions in living organisms. Some transport nutrients across the cell membrane, some fight infectious organisms, some are hormones, some are enzymes, etc. Collagen is the most abundant protein in the animal world and the ribulose, biphosphate, carboxylase, oxygenase means rubisco is the most abundant protein in the whole biosphere. 9.5 Polysaccharides. The acid insoluble pellet also has the polysaccharides, that mean carbohydrates, as another class of macromolecules. Polysaccharides are long chains of the sugar. They are threads, means literally a cotton thread containing different monosaccharides as a building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is variant of this but present as a storehouse of energy in a plant tissues. Animal have another variant called glycogen. Insulin is a polymer of fructose. In the such polysaccharide chain, say glycogen, the right end is called the reducing end and the left end is called the non-reducing end. It has the branches as shown in the form of cartoon. Figure 9.2. Starch forms the helical secondary structures. In fact, starch can hold I2 molecules in the helical portion, and the starch I2 is blue in color. Cellulose does not contain the complex helices and hence cannot hold the I2. Plant cell walls are made up of cellulose. Paper made up of from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars. For example, glucosamine, N-acetyl galactosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. 9.6. Nucleic acids. The another type of macromolecule that one would find in acidic insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with the polysaccharides and polypeptides, these comprise the true macromolecular fraction of the any living tissue or a cell. For nucleic acid, a building block is a nucleotide. The nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is the monosaccharide, and third is a phosphoric acid or a phosphate. As you notice in figure 9.1, the heterocyclic compound in a nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine and thiamine. Adenine and guanine are substituted purines while the rest are the substituted pyrimidines. The skeletal heterocyclic ring is called the purine and pyrimidine respectively. The sugar found in the polynucleotides is either ribose, means a monosaccharide pentose, or a 2-deoxyribose. A nucleic acid containing deoxyribose is called the deoxyribonucleic acid, means DNA, while that which contains the ribose is called a ribonucleic acid, RNA. 9.7. Structure of proteins. Proteins, as mentioned earlier, are the heteropolymers containing strings of amino acids. Structures of molecules means different things in a different contexts. In inorganic chemistry, the structure is invariably refers to the molecular formulae, for example, NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of a molecular while representing the structure of molecules. Example, benzene, naphthalene, etc. Psychists conjure up the three-dimensional views of a molecular structures while biologists describe the protein structure at the four levels. The sequence of an amino acid, that is the positional information in the protein, which is first amino acid, which is second, and so on, is called the primary structures. Figure 9.3a of the protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called the N-terminal amino acid. The last amino acid is also the C-terminal amino acid. A protein thread does not exist throughout as the extended rigid rod. The thread is folded in the form of helix, similar to the revolving staircase. Of course, only some portions of the protein thread are arranged in a form of helix. In proteins, only right-handed helices are observed. Other regions of the protein's threads are folded into the other forms in what is the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Some proteins are the assembly of more than one polypeptide or subunits, the manner in which these individual folded polypeptides or subunits are arranged with respect to each other. For example, linear string of a spheres are arranged one upon each other in the form of cube or a plate, etc., is the architecture of the protein, otherwise called a quaternary structure of the protein, that is figure 9.3d. Adult human hemoglobin consists of 4 subunits. 2 of these are identical to each other, hence 2 subunits of alpha type and 2 subunits of beta type together constitute the human hemoglobin, that is Hb. 9.8 Enzymes. Almost all enzymes are proteins. There are some nucleic acid that behave like an enzyme. These are called the ribozymes. One can depict an enzyme by a one-line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and the tertiary structure. When you look at the tertiary structure, upon itself, the chain crisscrosses itself and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is the crevice or a pocket into which the substrate fits. Thus, the enzymes through their active sites catalyze reactions at the higher rate. Enzyme catalysts differ from the inorganic catalyst in many ways, but one major difference needs a mention. Inorganic catalysts work efficiently at the higher temperatures and high pressures, while enzymes get damaged at the high temperatures, say above 40 degrees Celsius. However, enzymes isolated from the organisms who normally live under extremely high temperature, example hot vents and sulfur springs, are stable and certain their catalytic power even at a high temperatures up to 80 degrees to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from the thermophilic organisms. 9.8.1 Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compound undergo two types of changes. A physical change simply refers to the change in a shape without breaking of bonds. This is a physical process. Another physical process is a change in a state of matter when ice melts into water or a water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during the transformation, this will be called as a chemical reaction. For example, BaOH twice plus H2SO4 means sulfuric acid gives BaSO4 and 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of a starch into glucose is an organic chemical reaction. Rate of a physical or a chemical processes refers to the amount of product formed per unit time. It can be expressed as rate is equals to delta P upon delta T. Rate can be called velocity if the direction is specified. Rate of the physical and the chemical processes are influenced by the temperature among other factors. A general rule of thumb is that the rate doubles or decreases by half for every 10-degree change in either direction. Catalyzed reaction proceed at the rates vastly higher than that of the uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but the uncatalyzed reaction. For example, CO2 means carbon dioxide plus H2O means water. Carbonic anhydrase as a catalyst gives H2CO3 means carbonic acid. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called the carbonic anhydrase, the reaction speeds dramatically with about the 6 lakh molecules being formed every second. The enzyme has accelerated in the reaction rate by the 10 million times. The power of enzymes is incredible indeed. There are thousands of types of the enzymes, each catalyzing a unique chemical or a metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called the metabolic pathway. For example, glucose gives 2-pyruvic acid, means if we add the O2 in the glucose, it gives the 2-pyruvic acid molecules plus 2 molecules of H2O. Is actually a metabolic pathway in which the glucose becomes the pyruvic acid through the 10 different enzymes catalyzed metabolic reaction. When you study the respiration in the chapter 12, you will study these reactions. At this stage, you should know that this very metabolic pathway with the 1 or 2 additional reactions give rise to variety of metabolic end products. In our skeletal muscle, under the anaerobic condition, lactic acid is formed. Under the normal aerobic condition, pyruvic acid is formed. In yeast, during the fermentation, the same pathway leads to the production of ethanol, means alcohol. Hence, in different conditions, the different products are possible. 9.8.2 How do enzymes bring about such high rates of chemical convergence? To understand this, we should study the enzyme a little more. We have already understood the idea of the active site. The chemical or metabolic conversion refers to the reaction. The chemical in which is converted into the product is called a substrate. Hence, enzymes that proteins with the three-dimensional structure including an active site convert a substrate S into the product P. Symbolically, this can be depicted as S into P. It is now understood that the substrate S has to bind the enzyme at the active site within the given cleft or a pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of ES complex. E stands for enzyme. This complex formation is an transient phenomenon during the state where the substrate is bound to the enzyme active site. A new structure of the substrate called the transition state structure is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of the substrate gets transformed into the structure of product S, product or products. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implied in this statement, in fact, that all the other intermediate. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this pictorially through the graph, it looks like something as in the figure 9.4. The y-axis represents the potential energy content. The x-axis represents the progression of the structural transformation or the states through the transition state. You should notice two things. The energy level difference between the S and the P. If P is at the lower level than the S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is the exothermic or the spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher energy state of transition state. The difference is an average energy content of S from that of this transition state is called the activation energy. Enzymes eventually bring down this energy barrier, making the transition of the S to P more easy. 9.8.3 Nature of the enzyme action. Each enzyme E has a substrate S binding site in its molecule, so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into its products P and the unchanged enzyme with a intermediate formation of the enzyme product complex EP. The formation of a ES complex is essential for catalysis. E plus S forms the ES complex, then gives the enzyme EP and gives the E plus P. The catalytic cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of the enzyme fitting into the active site. Second, the binding of the substrate induces the enzyme to alter its shape fitting more tightly around the substrate. Third, the active site of an enzyme now is close proximity of the substrate breaks the chemical bonds of the substrate and new enzyme product is formed. 4. The enzyme release the products of the reaction and the free enzyme is ready to bind the another molecule of a substrate and run through the catalytic cycle once again. 9.8.4 Factors affecting the enzyme activity. The activity of an enzyme can be affected by a change in conditions which can alter the tertiary structure of the protein. These include the temperature, pH, change in a substrate concentration or binding of a specific chemicals that regulate its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys the enzymatic activity because the proteins are denatured by a heat. Concentration of a substrate. With the increase in a substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in a concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after the saturation of these molecules there is no free enzyme molecules to bind with the additional substrate molecules. The activity of the enzyme is also sensitive to the presence of specific chemical that bind to the enzyme. When the binding of a chemical shuts off the enzyme activity, the process of inhibition and the chemical is called the inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as the competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for a substrate binding site of the enzyme. Consequently, the substrate cannot bind. As a result, the enzyme action declines. Example: inhibition of the succinic acid succinic dehydrogenase by the malonate, which closely resembles the substrate succinate in the nature. Such competitive inhibitors are often used in a control of a bacterial pathogens. 9.8.5 Classification and nomenclature of enzymes. The thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the types of reactions they catalyze. Enzymes are divided into 6 classes, each with the 4 to 13 subclasses and named accordingly by a 4-digit number. Oxidoreductases or dehydrogenases, enzymes which catalyze oxidoreduction between the two substrates S and S-. Example: S-reduce and S-oxidized gives the S-oxidized and S-reduce. Transferases, enzyme catalyzes a transfer of a group G other than the hydrogen between a pair of a substrate S and S-. Example: Sg plus S- gives S plus S-G. Hydrolases, enzymes catalyzing hydrolysis of a ester, ether, peptide, glycosidic, C bond C and C halide or PN bonds. Lysases, enzymes that catalyze removal of groups from the substrates by mechanisms other than the hydrolysis, leaving double bonds. Isomerases include all enzyme enzymes catalyzing interconversion of optical, geometric and positional isomers. Ligases, enzymes catalyzing the linking together of two compounds. Example, enzymes which catalyze joining the CO, CS, CN, PO, etc. bonds. Means carbon oxygen, carbon sulfur, carbon nitrogen and phosphorus oxygen, etc. bonds. 9.8.6 cofactors. Enzymes are composed of the one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called the cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzymes is called the apoenzyme. Three kinds of cofactors may identified: prosthetic groups, coenzymes and metal ions. Prosthetic groups are the organic compounds and are distinguished from the other cofactors in that they are tightly bound to the apoenzyme. For example, the peroxidase and catalase, which catalyze the breakdown of a hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it is a part of the active site of the enzyme. Coenzymes are also the organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of a catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example, coenzyme nicotinamide adenine dinucleotide means NAD and NADP contain the vitamin niacin. A number of enzymes require metal ions for their activity, which forms the coordination bonds with side chains at the active site and at the same time form one or more coordination bonds with the substrate. Example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies they play a crucial role in a catalytic activity of the enzyme. Summary: although there is a bewildering diversity of the living organisms, their chemical compositions and metabolic reactions appear to be remarkably similar. The elemental composition of living tissues and non-living matter appear also to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen, oxygen is higher in the living systems. When compared to inanimate matter, the most abundant chemical in a living organisms is water. There are thousands of small molecule weights lesser than 1000 Dalton biomolecules. Amino acids, monosaccharides and disaccharides, sugars, fatty acids, glycerol, nucleotides and nucleosides, nitrogenous bases are some of the organic compounds seen in the living organisms. There are 20 types of amino acids and 5 types of nucleotides. Fats and oils are glycerides, in which the fatty acids are esterified to glycerol. Phospholipids contains, in addition, a phosphorylated nitrogenous compound. Only 3 types of macromolecules, that is, proteins, nucleic acid, and the polysaccharides, are found in the living systems. Lipids, because of their association with the membranes, separate in the macromolecular fraction. Biomacromolecules are polymers. They are made up of building blocks which are different proteins are heteropolymers made of the amino acids. Nucleic acid, RNA and DNA are composed of nucleotides. Biomacromolecules have a hierarchy of structures: primary, secondary and quaternary. Nucleic acids serve as a genetic material. Polysaccharides are the components of a cell wall in plants, fungi and also of the exoskeleton of the arthropods. They also are the storage form of energy. Example, starch and glycogen. Proteins serve a variety of cellular functions. Many of them are the enzyme, some are antibodies, some are receptors, some are hormones, and some others are the structural proteins. Collagen is the most abundant protein in the animal world, and ribulose biphosphate carboxylase oxygenase means Rubisco is the most abundant protein in the whole biosphere. Enzymes are the proteins which catalyze the biochemical reactions in the cells. Ribozymes are nucleic acid with the catalytic power. Proteinaceous enzymes exhibit the substrate specificity, require the optimum temperature and the pH for the maximal activity. They are denatured at the high temperature. Enzymes lower activation energy of the reactions and enhance greatly the rate of reactions. Nucleic acid carry hereditary information and are passed on the parental generation to progeny. Thank you.